The title of our session is Pathophysiology and Potential Mechanisms of ARIA. We have a total of 6 speakers, 1 virtual, number 4, and the rest of the speakers are here in person. And we will go very quickly down the line. We'll be giving, we will have talks from Constantino Ayatacola, then Delphine Bosch, then me, and then Praveen Battini, who is a postdoc in my lab, and he'll be speaking virtually, and then Donna Wilcock, and finally wrapping it up with Xavier Taylor. And so we're looking forward to lots of questions during our discussion period. I am asked to tell you that this session is being offered as a continuing medical education and continuing education course. For APA credit, there will be an iStart ambassador who has the sign-in sheets available at the back of the room. If you have any questions, please visit the AAIC Continuing Education webpage or visit the Continuing Medical Education registration desk near the registration area. Okay, and so with that, without further ado, I'd like to introduce our first speaker, Professor Constantino Ayatacola from Weill Cornell Medical School.
Thank you, Cindy. Okay, so I can get to the microphone. All right, come forward because this is a very big room for this talk. So we're going to start from the very beginning. So from the cerebral vasculature, because ARIA is a vascular problem. So the vessels of the brain have many functions, not just the delivery of blood flow to the neurons who need activity, and because of their activity, they need energy. So one of the major factors is called neurovascular coupling, which is the ability of neural activity to increase blood flow in measure of the energy being used by the neurons themselves. So that's one of the functions. Another function rests with the endothelial cells, which is the lining of cerebral blood vessels, which are responsible for regulating microvascular flow, as well as the blood-brain barrier, which is the exchange of compounds between the blood and the brain. But then, most recently, another aspect of vascular function has become obvious, which is the role of the vessels in getting rid of the waste that the brain generates during neural activity. So neural activity and glial activity generates a bunch of things that the brain doesn't want to keep around, like, for example, A-beta and tau that need to be removed. And all of the vessels contribute to this removal because all this kind of junk, quote unquote, get to the perivascular space around the blood vessels, and from there is removed from the brain through a anterograde or retrograde pathways. You may have heard of the lymphatic pathways. You may have heard of the IPAD system, retrograde propagation along the perivascular space and reaching the subarachnoid space. Eventually, the stuff gets out of the brain, no matter how, but the vessels are critical in these functions. That's another very important function of the vessels. Another very important function is the immune privilege of the brain. So the brain vessels are closely associated with immune cells. They continuously patrol the perivascular space and the meninges. So these are both adaptive immune cells like T cells. They're also innate immune cells, as we will see, like macrophages, monocytes, and so on. So these are critical to maintain the integrity of the brain, the immune integrity of the brain. And they go back, with these cells go back to the periphery to tell the immune system what is going on. And then they come back and they establish this so-called immune privilege of the response of the brain. And finally, the vessels of the brain also generate trophic factors that keep neurons and glia alive. So all these things are not just delivering the blood to the brain, the plumbing, but it's also these other functions which are very important for brain health. And so it's not surprising that if these things don't work right, all right, you're going to have a problem. And often the problem is cognitive dysfunction. And so a number of factors that profoundly affect vascular functions are also associated with cognitive impairment. And one of the critical mechanisms whereby these factors that cause cognitive impairment, like amyloid beta, like ApoE, and like hypertension, rest with innate immune cells closely associated with cerebral blood vessels. These are called perivascular macrophages. These are a class of the myeloid cells that enter the brain very early in development, and they divide to give up the four major classes of myeloid cells in the brain: the microglia, which we all know and love, as well as perivascular macrophages, meningeal macrophages, and choroplexus macrophages. So these cells, particularly the perivascular macrophages, are loaded with free radical producing enzymes and with cytokines and other pro-inflammatory mediators. So it's like having an atomic bomb stuck to a blood vessel. So when these cells are activated, they cause havoc on the blood vessels. And that's what happens, for example, during amyloid accumulation. A-beta binds to innate immunity receptors called CD36, as well as other immunity receptors in the macrophages, leading to production of free radicals and oxidative stress and inflammation through enzymes that these cells are enriched with. And then this leads to the whole slew of neurovascular problems that we see. So there is neurovascular uncoupling. The neurons want blood, but they don't get it. All right? There is endothelial dysfunction, so the endothelial cells are no longer able to regulate blood flow. There is blood-brain barrier alteration, and there is failure of the clearance mechanisms. All these things contribute to this deleterious effect of A-beta. Similarly, ApoE4, which is a major risk factor for both vascular cognitive impairment and Alzheimer's disease, is able to, through a similar mechanism, but to different receptors, to produce neurovascular dysfunction. In this case, ApoE4 is produced by the macrophages themselves, and they act on the macrophages in a cell autonomous way to activate a receptor for ApoE4, leading to, again, oxidative stress and inflammation and whole neurovascular dysfunction problem. Similarly, hypertension is able to alter neurovascular function also by activating receptors in the macrophages, either IL-17, which is very commonly associated with the human hypertension, or angiotensin II, another major factor in human hypertension. And these are able to activate the cells, leading to neurovascular dysfunction. Now, you may wonder, what does this have to do with A-beta immunotherapy? So A-beta immunotherapy, is, as you know, is the first disease-modifying treatment from Alzheimer's disease. As you can see on this side here, you can see the effect, the positive effect of delaying the cognitive impairment and completely, or nearly completely, clearing the A-beta accumulating in the brain, so acting on one of the pathogenic factors on Alzheimer's disease. However, this wonderful achievement has got a problem, which is shown by the next slide, which is this phenomenon ARIA. So ARIA, amyloid-related imaging abnormality, is an imaging phenomenon that comes in two flavors: area E, which means area edema, which is characterized by accumulation of water essentially in the brain. The neuroradiologists call this, vasogenic edema due to vascular dysfunction, and accumulates and causes swelling of the brain. And the other one is called area H, where you can have microhemorrhages as well as superficial siderosis, which is a little bit of blood seeping out of the peel surface of the brain. So most often, the ARIA is asymptomatic. However, in some cases becomes symptomatic, and it can be characterized by headache, confusion, and other alteration. However, in the setting of vascular diseases, particularly stroke, cases of ARIA turn to be fatal. So ARIA can be very, very serious and needs to be obviously looked at with importance because it's critical. And so what happened to the brain when you give these antibodies to clear the A-beta? Well, the antibodies do a great job at getting rid of the plaques. As shown in this slide, you can see a control brain and a brain treated with aducanumab when the patient was alive. However, the CAA, the cerebral amyloid angiopathy, the accumulation of amyloid around the blood vessel gets worse. So it's like moving the amyloid from the parenchyma to the blood vessel. And obviously, this is not good for the blood vessels. Now, Interestingly, the major risk factors for ARIA that have emerged so far, so by this, the field is not complete by any means, are essentially the same factors that I told you before: ApoE4, cerebral amyloid angiopathy, and hypertension. All of them contribute to have this profound, to increase the risk of ARIA by some from several fold. So if we want to put all this stuff together, then we can say that when the amyloid, the antibodies are administered, the plaques are cleared, some of the A-beta is going to be metabolized by the microglia, some of it is going to escape along the physiological clearance route, which brings this amyloid to the blood vessels, engaging the perivascular receptors and the perivascular macrophages. Now, if the perivascular macrophages are already primed to be in an inflammatory mode, or by hypertension, by ApoE4, or by a pre-existing accumulation of A-beta around the blood vessels, then the thing gets much worse, and you have essentially vascular failure, resulting in not only in edema, but also in exit of blood vessels, of red blood cells from the blood vessels. Other factors which may play a role, and will be discussed in detail by my colleagues here, involve the role of the complement and immunocomplexes that may activate these immune cells further, in addition to the risk factors that I told you before, and also the fact that A-beta may be cleared also from the vascular wall, resulting in a weakening of the vascular wall. The role of perivascular immune cells in ARIA is really demonstrated by pathological studies showing CD163 positive cells, which are the perivascular macrophages, hugging the blood vessels and loaded with A-beta, presumably causing the vascular dysfunction. So in conclusion, ARIA is essentially a neuroimmune microangiopathy. And amyloid, hypertension, ApoE4 may increase the risk by priming the macrophages to be in a hyper-inflammatory state. And therefore, if you want to use immunotherapy correctly, you have also to think about the neurovasculature. And this may result in mitigation of the vascular risk associated with ARIA, as well as with a better outcome of the A-beta immunotherapy. So thank you very much. So now we move.
I think Delphine is next.
Oh, you want to? Okay.
Sure. So our next speaker, thank you very much, Cas. Our next speaker is Professor Delphine Bosch from University of Southampton in the United Kingdom, and the title of her talk is ARIA, Insights from Human Neuropathology. And before we get started, please think about your questions. We're going to have the 6 talks in a row, and then we'll have a half hour for discussion. Thank you.
And good afternoon, and thank you to the organizer for the opportunity to present evidence from the human neuropathology of the potential underlying mechanisms of ARIA. So here's my disclosure. I have no financial interest in relation to Alzheimer immunotherapy. So as this has been just described, so amyloid-related imaging abnormality, or ARIA, actually has been described for the first time with the bapineuzumab. There's 2 types of ARIA, E for edema, H for hemorrhage. And as has been mentioned, some risk factor has been identified, so being homozygote for the ApoE4 or the pre-existing of microbleed. What is quite interesting is actually ARIA was always observed with the higher dose of antibody and was localized with the reduction of amyloid as observed on PET scan. ARIA-E is restricted to weeks and months after onset of treatment and is reversible. And what it was quite interesting is actually that ARIA had some similarity with the AN72 meningoencephalitis, which has been described years before. So the AN72 clinical trial was an active protocol with a full length of A-beta 42, and it was a 2-year protocol performed in the United Kingdom. So what we've done in Southampton was a long-term clinical and neuropathological follow-up of the patient. So what we did observe, so this is the first postmortem examination of an immunized patient, and it was clear that there was a striking A-beta removal from the cortex compared to an unimmunized patient. So to date, we had 22 brain, and what we were able to observe was the high variability in the pattern of A-beta removal, from very extensive removal to no removal, and between these there was a degree in the A-beta removal. So we perform a quite detailed neuropathological examination of the different changes, and these changes were still observed 14 years after the A-beta immunotherapy. So one of these observations was that there was a clear removal of A-beta in the parenchyma, and this was associated with an increase of the CA. And here there is a bit of a kinetic of event, if you want, so looking at different brain at different times. So here it's an immunized AD, which has been stained for A-beta. 4 months later, we can already see some appearance of A-beta removal. We describe that as the mossy-eaten plaque. And then months later, with what we observe in an area which has been clear of plaque, it's this presence of severe CA. And then years later, some of the cases didn't have any more A-beta. This was associated with the presence of hemorrhage, so identified with the pulse staining. And on H&E, we were able to observe some microvascular lesion. We also describe a CA-associated vasculopathy, so which is this increased vessel wall split, as you can observe here, after the A-beta immunization. And we were wondering whether this can actually reflect the removal of A-beta from the vessel over time. So 2 cases were published, so one from the first clinical trial, the second one by Isidro Ferrer from the second clinical trial, the one which was halted 6 months later, and it was described as a meningoencephalitis. So what we can observe, looking at the brain imaging, is actually what we observe is an ARIA E, and this is characterized as well by the presence of lymphocytes in subarachnoid space, which cluster around the artery, with a double barrel morphology associated with a severe CA. We've got this white matter rarefaction observed with the Lux-sulfase blue, and we can observe old cortical microbleed with a cluster of hemosiderin pigment granules. And what was quite striking, it was a severe CA in leptomeningeal, but also in parenchyma. So when we look at a bit of a larger view on the brain, so what we were able to observe was actually the severe CA was in the leptomeninges, and the parenchyma was associated with an area which was clear of the A-beta plaque. And then there was the presence of a cortical microbleed, severe CA with inflammation. And in the white matter, we were able to observe the enlarged perivascular space, which is usually a sign of edema. So a few years ago, actually, this was published in 2010, we suggested these pathophysiological mechanisms for ARIA. So we do have the plaque. Then following the A-beta immunization, we've got the antibodies that will bind the A-beta in a plaque. These will solubilize the plaque, and then the soluble A-beta will be either phagocyted by the microglia, but also drained towards the blood vessel. And then this will be associated with an increase of CA. And this will lead to 2 events. The white matter abnormality, so which is maybe an altered fluid balance and associated with vasogica edema, which might be due to impaired vascular drainage or ischemia. And this edema is more common in people with the ApoE4, because actually people with ApoE4 are much more prone to have already CA in their blood vessel. And interestingly, this was also associated with the rapid plaque removal. And then the second event is the removal of A-beta from the vasculature, potentially by the antibody, and this will create an inflammatory response with the presence of T cells on macrophage. It will fragilize the blood vessel and will lead to microhemorrhage. So in collaboration with Suzanne van Veloos, she used the ex vivo magnetic resonance imaging on some of our immunized Alzheimer's case. So here actually the 1021 is a case with a meningoencephalitis. And what she observed was that the hemorrhagic lesion on the ex vivo MRI correspond to the presence of iron and calcium, and that the deposit of iron, which is a sign of the hemosiderin just mentioned previously, was associated with lower A-beta plaque area in the immunized AD cases. So still we have this association between the presence of hemorrhage, the absence of A-beta plaque. More recently, has been also reported, some patient died after treatment with lecanemab. So here it's one of the patients. So he was a 65-year-old with cognitive decline. He was homozygote for the ApoE4 and was treated for lecanemab. 4 days after the last infusion, he developed ischemic stroke and therefore was treated with a TPA. As a result, he developed multiple hemorrhage. So what is not clear here in the event related to the death of the patient is whether there is a role for the CA, a role for the ApoE4, a role for immunotherapy, a role for the treatment for the stroke, or is it actually a result of the combination of these different events? A second patient was a 79-year-old patient who was also homozygote for the ApoE4 and was also treated for lecanemab. This patient was also treated for atrial fibrillation with heparin. The patient developed multiple cerebral hemorrhage and presented with CA-related inflammation. So similarly as the first patient, it's unclear what led to the cause of the death of the patient, whether it's a role for the CA-related inflammation, the role for ApoE4, for the anticoagulation, or the immunotherapy, or whether it's a combination of all these events. So the key point from the neuropathological assessment of the patient who were immunized against A-beta from the first clinical trials, the ARIA in 1792. So ARIA seems to be due to dynamic changes in the location of A-beta, with a shift from plaque to vessel wall, which is associated with the increase of the CA. But then over time, A-beta can be also removed from the vessel walls, and there will be an inflammatory response associated with this removal. ARIA-H is likely due to the damage to the integrity of walls of small artery and arteriole in cortex and leptomeninges, which is affected by CA. ARIA-E is likely due to the failure to control water flux at the blood-brain barrier as a result of vascular pathology. So the risk factor for developing ARIA are to carry on the E4 for the ApoE and the pre-existing of CA. So the advantage of the passive immunotherapy protocol is that it permits monitoring of ARIA by imaging and therefore to lead to the reduction or temporary or even maybe permanent withdrawal of the treatment. Question related to concomitant thrombolysis, anticoagulation, or any treatment which might affect the blood vessel is actually an important question. At the moment, there is some limitation to really understand the physiopathomechanisms of ARIA, partly due to the difficulty to study ARIA because there is really few tissue available for study. But however, interestingly, ARIA seems to present some analogy with spontaneous disease of cerebral amyloid-related inflammation. Now, maybe one of the key questions, if active protocol was going to be used as a prevention of Alzheimer's disease before the patient developed a large amount of A-beta and before the development of tau, maybe we will be able to prevent the disease without the occurrence of ARIA. So a number of collaborators over the year has been involved in this study of this immunized cohort, which is quite unique worldwide. Two main collaborators in Southampton were Professor Clive Holmes and Professor James Nichol. I would like to thank our founders, the UK RISE and the Alzheimer's Research UK, the UK Brain Bank Network, and also the patient and relative who allows this study. Thank you.
So our next speaker is going to be Cindy Lemire from Harvard, and she's going to talk about.
I will be talking about animal models, mice in particular, as tools for studying ARIA. So hopefully we can have my slides. May I have my slides up? Thank you very much. Okay. These are my disclosures. So you've already heard from both Cos and Delphine about some of the risk factors contributing to ARIA, and this includes ApoE4 genotype, abundance of amyloid, and in particular CAA or vascular amyloid, pre-existing microhemorrhages or white matter disease, dose, higher doses tend to be more inducing of ARIA, hypertension, antithrombotics, and age. So all of these can be mimicked in mice and studied. I'm going to focus mostly on CAA today, but there are a number of other studies that are ongoing now to look at some of these other factors, such as hypertension, which is something Sally Froschi is doing at USC right now. So was ARIA predicted by preclinical studies in animal mouse models? And the answer to that would be yes. I'm going to see if I can get this to work. There we go, I think. So on the left, this is basically, these are 3 different figures from 3 different papers showing very early on that, thank you, very early on that ARIA-H at least was present in these different mouse models in old mice with CAA present at the time of immunization. And so on the left, you've got an N-terminal antibody used in an APP23 mouse model, and these mice do develop spontaneous microhemorrhages even without anti-amyloid immunotherapy, but the amount of microhemorrhage goes up with treatment. In the middle, we've got a paper from Donna Wilcock showing that in TG2576 mice using a C-terminal antibody at 19 to 23 months of age, you can see that this induced microhemorrhages in the cortex, and it was associated with increased microgliosis. And then on the right, Raki et al. Showed in 22-month-old PDAPP mice that 3D6, this is the bapineuzumab murine precursor antibody, and that reliably induced microhemorrhages in these old PDAPP mice. On the left, you can see a paper from Dora Games and Sally Schroeder from Milan showing that there's a dose-dependent effect. So the more 3D6, the higher the amount of microhemorrhages. And in particular, these were PDAPP mice treated starting at 12 months of age, but they were treated weekly for 6 months. So that's 24 treatments. And these mice ended up having quite a bit of microhemorrhages. Another study from our final speaker in this session, Xavier Taylor from Eli Lilly, showed that in roughly 2-year-old PDAPP mice, weekly treatment with 3D6 for 3 months led to abundant microhemorrhages. And for the most part, these are along the surface of the brain and the leptomeninges and in these large penetrating arterioles. And then Terry Boussier and Ed Plowry at Biogen used imaging to actually see both ARIA-E, shown in A and B, and ARIA-H in 5XFAD mice. 5XFAD mice, this is the line that's available from Jackson Laboratories, 6799. They do develop some CAA. It's not particularly robust, but they do develop some CAA with aging. And here he showed that there is immune cell accumulation along the leptomeninges. There's albumin staining, which is indicative of a fusion in the cortex, and then Pearl's iron staining, which is indicative of microhemorrhages. And then there's a mouse model called RT10, available from Jackson Lab, and this mouse model has been shown to have PIB binding, so this is tritiated PIB that was used to stain brain sections, and you can see lots of plaques, but you can also see these blood vessels, again, large penetrating arterioles, that were PIB positive, and in addition, were both A-beta 40 and 42 positive at 2 years of age. Also at 2 years of age, this group at University of Rhode Island, Ken Rose and Katie Dunton, they also are part of Mind Immune Therapeutics. You can see that these mice have quite a bit of vascular amyloid stained by a dye called AmyloGlow, and they also, with 3D6 treatment, develop some microhemorrhages. Now, this paper just came out very recently from AbbVie, and they used an APP-PS1-21 mouse model, and they treated these mice at 15 to 17 months of age. They gave them one single bolus of 40 mg per kg antibody. That's a lot of antibody. They euthanized the animals 72 hours later, and what they found was that unlike their pyroglue antibody that does not seem to induce ARIA, they did see this increase in ARIA. This is red blood cells accumulating along the surface of the brain with 3D6 treatment, and quite a bit of microhemorrhage along the surface and the penetrating arterioles, again with 3D6 antibody. And this is work from our lab. Praveen Bettini is a postdoc in the lab heading these studies, and he'll be the next speaker. But you can see that 3D6, which is a very, very reliable antibody for inducing microhemorrhages, it actually can see some of the hemorrhages on the surfaces of the brain. By MRI imaging, we could see some microhemorrhages, and then the same mouse brain was then histologically examined with Prussian blue, and you can see accumulation in the same areas that showed up on the MRI. And this is just showing a close-up of the amount of microhemorrhage in the penetrating arterioles. So the question is, are there any faster amyloid mouse models? People ask me all the time, can you recommend a mouse model that doesn't take 18 to 24 months to develop CAA? And so there are several lines out now. This is one that we're using. This is the 5XE4 line from David Holtzman. And I just want to point out here, this is the 5XFAD mouse model, but on a different background. So this is not a different background. It's a different line 7031. And these mice have a very low expression level of the APP and presenilin trans genes compared to the commercially available line at Jackson Laboratory. These mice were put onto a humanized ApoE4 knock-in background and by Dave Holtzman's lab, and with funding from Cure Alzheimer Fund, so you can reach out to them if you want to get this model. And these mice were treated weekly from 8 to 10 months of age with with chimeric aducanumab, and you can see that there's Prussian blue staining that co-localizes with vascular amyloid in these mice. And again, they saw this increase in microhemorrhages with chimeric aducanumab, but not with their ApoE antibody. These are some images from our lab now. This is one of the breeder mice that we got that has reached 10 months of age, and with AmyloGlow, you can see this abundant vascular labeling of these penetrating arterioles. And along the leptomeninges and penetrating arterioles, we do see Prussian blue or iron deposits indicating that 3D6 resulted in microhemorrhages. There are a couple of other mouse models that I can recommend. One is the WSB mouse model on the APP Swedish PS1 delta E9 mice. This is from Jackson Laboratory. They looked at wild-derived strain differences in the APP PS1 model, and they found that 92% of the mice on the WSB background had vascular amyloid. And they also saw that this vascular amyloid was related or associated with fibrin staining, which would indicate some compromise of the vascular integrity or interruption of the blood-brain barrier. There are two newer Riken mouse models. This is the NLF knock-in paired with a presenilin knock-in. So these are double knock-in mice. They express physiologic levels of each protein, and these animals show vascular amyloid. This is at 12 months of age, and this is with an ASP1 N-terminal antibody. So they do have vascular amyloid at 12 months. We're now looking at earlier ages to see exactly when they start to develop vascular amyloid. And then last but not least, there's this paper from Tachida et al. In which they overexpressed, or they didn't overexpress, sorry, they used a Cree model to specifically express human APP 770 in endothelial cells, and then they crossed those mice to an APP knock-in, APP NLF knock-in, and they found that these animals did have an increase in vascular amyloid. And both of these mouse models are available from Riken. So in conclusion, CAA appears to be required for ARIA in amyloid mice, and it looks like in humans. Dose anti-amyloid antibody and frequency of treatment seem to matter. Despite regional differences in CAA in amyloid mice, for example, some mouse models have Most of the CAA in cortex, others have a lot in cerebellum, or some animals have it in both. Chronic treatment with plaque-binding antibodies induces microhemorrhages in these animals. Red blood cell extravasation, blood-brain barrier leakage, and MRI detection of ARIA-E demonstrates that this can also occur in mice, and it often precedes microhemorrhages. These newer mouse models, which develop CAA earlier, may save time and money, and there's also a need to look at some of the other risk factors mentioned on the first slide. Understanding the temporal changes during anti-amyloid antibody treatment may help identify mechanisms, targets for mitigation of ARIA, and biomarkers. And with that, I'd like to just thank my lab and all of our collaborators. Thank you.
So the next presentation is going to be virtual. And it's going to be by Praveen Bhattini from Dr. Lemaire's lab, and it's going to be entitled Complement-Mediated Vascular Inflammation Contributes to Anti-Amyloid-Induced ARIA in Mice.
Good afternoon, everyone. I'm Praveen Bhattini, a postdoc from Lemire Lab, Brigham and Women's Hospital, Harvard Medical School. I thank the organizing committee for providing me this opportunity to share our recent findings on the role of complement cascade in anti-amyloid monoclonal antibody-induced ARIA. As a background, you all may know Alzheimer's disease is characterized by the extracellular A-beta plaque deposition as well as intracellular taut tangles. Importantly, A-beta also gets deposited in the cerebrovasculature, a condition called cerebral amyloid angiopathy or CAA. 3 anti-A-beta monoclonal antibodies have been approved by FDA, which include aducanumab, lecanemab, and donanemab. Aducanumab, however, recently got discontinued, and currently we have lecanemab and donanemab for the early symptomatic AD treatment. However, these immunotherapies are associated with a vascular side effect called ARIA, and ARIA stands for amyloid-related imaging abnormalities, as depicted on the MRI scans shown below here. ARIA encompasses both vasogenic edema, where fluid leaks into the brain parenchyma causing edema, and hemorrhages characterized by the accumulation of hemoserine deposits due to the RBC cell lysis. In rare cases, about 1 to 1.5% of the treatment conditions, it can also result in fatal cerebral hemorrhages, like in this patient shown here treated with lecanemab. It is important to understand in this context the role of ApoE genotype and the number of infusions in relationship to ARIA. Aggregated data for aducanumab clinical trials showed increased risk for ARIA in patients having ApoE4, and in lecanemab and donanemab clinical trials, it was clearly shown that patients having 2 copies of ApoE4 were shown to have increased risk for ARIA versus patients having 1 copy of ApoE4 gene. ARIA most often appears early during the treatment, typically within the first few biweekly infusions in patients received lecanemab, or during the first few monthly infusions of donanemab infusions. This brings us to the critical question, what is the underlying mechanisms under ARIA, and what is the critical time window for the ARIA development? Histological data both from the animal mouse models as well as human AD specimens showed increased complement protein deposition near the CA-Ladin vessels, potentially affecting the vascular integrity. In this context of anti-A-beta immunization, we hypothesized that antibodies binding to the vascular A-beta could trigger the classical complement pathway, further increasing the complement deposition around the CA-Ladin vessels and the formation of membrane attack complex formation C5b9, causing vascular integrity loss and BBB breakdown. To test this, we use multiple amyloid mouse models to answer some of the questions we had in our lab. First, to understand where do all anti-A-beta antibodies bind in the brain. Following acute immunotherapy, we used aged APP-NLGF mice which harbor cerebellar CA pathology, and these mice were treated with either IgG step control or murine bapnismab precursor 3D6L antibody. And kindly note that bapnismab antibody was shown to cause ARIA in clinical trials. So mice when treated on day 1 and day 4 and euthanized on day 7 for subsequent pathological studies. Also, in aged APPPS1 mice with human target replacement for ApoE4, when given 7 weekly immunizations or 13 weekly immunizations to understand the role of complement activation during short-term and chronic anti-A-beta immunization, and also to study the influence in their ARIA development. So in our acute 2-dose study, the 25-month-old APP-NLG of mice having cerebral CA pathology when treated on day 1 and day 4 and euthanized on day 7, and when the brain sections were probed for general S97A beta as well as anti-mouse IgG2A to detect the injected monoclonal antibodies, we found that the antibody localized to the leptomeningeal vessels as well as vessels near the ventriculum. These vascular bound antibodies recruited complement C1q, indicating early classical complement activation. However, there were no overt microhemorrhages observed as seen on the Prussian blue staining, nor RBC extravasation on HNE. Further, quantification showed prominent vascular localization of antibodies during their early exposure window when immunostained with anti-mouse IgG2A as well as vascular collagen basement membrane marker collagen 4. This indicates that during early antibody infusions, antibodies preferentially bind to the CA vessels, at least in this context. Further, in our 7-week short-term study, the APPPS1 mice, when treated with 3D6L or IgG control for 7 weeks, we found that there is increased deposition of complement C1q near the plaques, as well as in the CA vessels in the leptomeninges, and increased immunoreactivity for IC3b in the leptomeningeal vessels. This caused increased frequency of microhemorrhages in the 3D6L treated mice in the leptomeningeal vessels as well as in small vessels in the brain parenchyma. And the data showed significantly increased number of microhemorrhages in the 3D6L treated mice. We also found that there is increased RBC extravasation in these mice as shown with the eosin counter stain. In a different sub-study, 20-month-old APPS1 mice, when probed for brain cerebellar RNA transcriptome, we found upregulation of many genes, including the ones that are related to complement pathway, like C3, C1q, and increased matrix metalloproteinases, markers related to astrogliosis, lysosomal proteins, as well as lectins, indicating innate immune activation. Further, to understand the effect of long-term immunization on the complement activation, the APPPS1 mice, when treated for 13 weeks, we wanted to understand what is their effect on A-beta pathology. And in 13 weeks of immunization, we saw lowering of A-beta 42, A-beta 40, and increased A-beta 42 by 40 ratio in the 3D6L treated mice. And we also saw a trend for CA lowering in the 3D6L treated mice, at least a partial vascular A-beta clearance. This could have led to increased microhemorrhages in these mice, as shown here with the positive Prussian blue spots in the leptomeningeal vessels, as well as some penetrating vessels into the brain parenchyma and other brain areas like cerebellum. As hypothesized, we found complete classical complement activation from the upstream C1q deposition near the CA vessels, as well as in the A-beta plaques. We also saw intermediate fragments of IC3b, and also the downstream increase of membrane attacks complex formation C5b9 in the CA Laden vessels, which could have potentially caused vascular impairment. When probed for the brain C3 levels, we also saw increased soluble C3 levels in the brain in the mice that were treated with 3D6L, and this correlated with the frequencies of microhemorrhages observed in these mice. Further, we were interested in understanding the role of perivascular macrophages in the context of C3 associated recruitment, and we found out that there is increased co-localization for C3 positive C206 co-localized areas, indicating C3 recruitment of the perivascular macrophages. We also saw increased astroglial immunoreactivity in the 3D6L treated mice. Further, we started exploring the role of immune cell infiltration and immune cell activation in different immunization protocols. Firstly, in this acute 2-dose study, although we didn't saw any immune cell recruitment, we saw increased CA-associated TREM2 activation. Although in a different mouse model, by 7 weeks, we saw increased accumulation of CD3 cells or CD8 T cells, as well as increased accumulation of lysic G neutrophils, or ones that were stained with neutrophil elastase marker for degranulation. So to conclude, our studies showed that the 3D6 antibody initially binds to the vascular amyloid and activates the classical complement cascade. Short-term treatment significantly increases complement activation, microhemorrhages, and RBC extravasation. Subsequent dosing on a long-term level lowers the amyloid levels, but increases brain C3 levels, which positively correlate with the microhemorrhages, and as well as recruitment for perivascular macrophages, which could have potentially lead to vascular inflammation and ARIA. So therefore, targeting complement proteins may lower ARIA risk. Ongoing studies indicated that activation of perivascular macrophages as well as infiltration of immune cells, particularly neutrophils and T cells from the periphery. Finally, I thank my PI Cynthia Lemmer for her kind support for all these experiments, and thank my colleagues, funding agencies, and collaborators. For recent updates on our project, please visit our poster 108832. Thank you.
Great. Well, thank you very much, Praveen. That was excellent. And our next speaker is Professor Donna Wilcock from Indiana University School of Medicine, and the title of her talk is Investigating the Role of MMP9 in Anti-Amyloid Immunotherapy-Associated ARIA.
Good afternoon, everyone. Thank you to Dr. Lemire and Dr. Ida Kola for organizing this session and inviting me to be a part of it. So I want to talk about some historic data, but then some new data that we have relating to a potential role of MMP9 in anti-A-beta immunotherapy-mediated ARIA. These are my disclosures. None of these relationships relate to what I'm going to talk about here. So I think over a number of years, it's been clear that there is a role for microglia in the clearance of amyloid due to anti-beta immunotherapy. These are a series of studies that were done in 2004 and then later in 2011, where we showed in a time course study when I was working with Dave Morgan that we see a transient increase in microglial activation with increasing dosing. So these mice are TG2576 mice that are aged, and we dosed weekly with an anti-beta antibody for 1, 2, or 3 months, and then we looked at the pathology of those mice. So what you can see in the top right there is CD45 positive microglia, where we saw the peak microglial activation with CD45 after 2 months of administration, but that microglial activation actually came back down at the 3-month dosing time point. However, if we look at Fc gamma receptor expression on microglia, we see that that is actually increased earlier in the process, even after just 1 month of antibody dosing, stays elevated at the 2-month of dosing, but then returned back down to control levels after 3 months of dosing. If you look at the graphs on the left, this is the same study. We approached Dave to see if he still had tissue in the freezer a number of years later, and he did. So we did some gene expression analysis, looking at different hallmarks of inflammation. So we examined IL-6, TNF-alpha, INOS or NOS2. We looked at TGF-beta, YM1, which is a chitinase-like protein, arginase 1. And what you can see is there's a lot of variability depending on which cytokine or immune-mediated data you're actually looking at. So IL-6 peaked very early after just 1 month of dosing, but then went back down, whereas if we look at TNF-alpha, that stayed elevated throughout the 3 months of antibody dosing. Some of the kind of repair-associated genes, like the chitinases and the arginases, actually were decreased by the anti-A beta immunotherapy. That's the bottom graph there. But we also saw some activation of TGF-beta, which I'll come back to later on. This is an interesting potential pathway. And then just at the top, you can see the time course of the plaque clearance. So we really didn't see significant plaque clearance until we got to 2 months of antibody dosing in these studies. Again, back to the point that Dr. Lemire was making, the papers from Pfeiffer and then later from myself, and then subsequent to our paper, the paper from RAC, all showed in different mouse models with different antibodies. If your mouse model has a lot of CAA and you use immunotherapy, you will see microhemorrhages. And as you can see, these papers are 2002, 2004, and later 2005. So I have this image from Dr. Lemire, but some interesting things that we know that have been identified as risk factors for ARIA in the clinical population. I think pre-existing microhemorrhages suggest that vasculature is already susceptible, and so that increases risk for ARIA. Obviously, ApoE4 being the biggest one, and we still don't really know if that ApoE4 risk is simply because ApoE4 individuals are known to have more CAA, or whether there's something else happening with ApoE4, because we also know that ApoE4 can alter the way that microglia respond to stimuli. The other, I think, interesting point that came out of some of these analyses is hypertension also seems to be a risk factor for ARIA, and that's something that clinically we've been implementing and monitoring. So the question, is there a role for MMPs in blood-brain barrier breakdown due to anti-A beta antibody? This is the same 3-month study that I showed earlier. In this case, we're looking at gene expression of various matrix metalloproteinases, which are known to degrade tight junction proteins, degrade proteins in the basement membrane, and in particular in stroke, MMP9 has been extensively studied as being a key factor in the hemorrhagic transformation after an ischemic stroke. So what we saw in our anti-A beta immunotherapy treated mice was we saw a significant increase in the expression of both MMP2, which is on the top graph there, and MMP9. We didn't really see the endogenous inhibitor increase, which would suggest that those MMPs are going to be more active. We focused in more on MMP9 because MMP9, the time course, so we didn't see an increase in 9 until we hit 2 months of dosing, and then it remained high at 3 months of dosing, and that is also the time course for when we see the microbleeds. So we don't see an increase in microhemorrhages until 2 months of dosing, and then it continues to increase with increasing numbers of antibody doses. We also saw an increase in MMP3 along the same time course, and it's important to note that MMP3 is necessary for the cleavage and activation of MMP9. So the fact that we saw both of these increased along that same time course suggested to us that there could be a role for MMP9 in the development of these microhemorrhages. So this is our working hypothesis is that when you have antibody bound to parenchymal plaques, you engage microglia, you get plaque clearance through phagocytosis, You have signaling to the astrocytes through inflammatory cytokines. We know that the astrocytes are the primary source of MMP9 in the brain, not the only source, but the primary source. And MMP9 can actually degrade A beta. So when this is happening around a parenchymal plaque, this may actually be part of the clearance mechanism and be beneficial. However, when the antibody binds to amyloid that is in the vessel as CAA, you engage the same pathway, but that activation and release of active MMP9 will degrade tight junction basement membrane proteins, leading to leakage of that vessel, which will lead to the vasogenic edema and microhemorrhage. So this is our working hypothesis. I will go through this quite quickly because this work was recently published in Journal of Neuroscience by my postdoc, Kate Foley. But we wanted to take some of what we'd learned back in the early aughts, where we didn't have single-cell RNA-seq technologies and that kind of thing, and go back and look at what microglia are actually doing once they see anti-A beta antibody in the brain. And so this was an acute study where we intracranially injected either 6E10, which is an N-terminal anti-A beta antibody, or an isotype-matched control antibody. And we euthanized the mice either 24 hours or 3 days after that injection, and taking the injected frontal cortex, performed single-cell RNA-seq, looked at, in particular, looked at communication networks using cell chat to look at what potential microglia populations are signaling to what other microglia populations. And also, through this preparation, we also have peripheral immune cell clusters that we were able to look at. So you can read the paper. Just to very quickly summarize, what we found was that the microglial signaling 24 hours after injection is incredibly diverse. We see lots of different microglial populations communicating with each other. There was very strong TGF-beta signaling detected just after 24 hours of anti-A beta antibody injection. What we saw at 3 days was a much more focused communication between a smaller number of microglial clusters. So I recommend you pull the paper from JNeuro and take a read at that. So coming back to our hypothesis, we wanted to examine what might happen if we inhibit MMP9 and give anti-A beta antibody. So this was our study design. I will tell you, we're still in the midst of analyzing data, so I'm just going to show you a little piece of data. So we're using the SAA knock-in mice that were produced through Model AD. So that's the humanized A beta. Normal sequence is the HAB, and that is our control mouse. They don't develop plaques. And then we have the HAB SAA, which are the ones who develop age-related plaque deposition. So what we did was we started them at 19 months, and they received weekly 3D6 injection. You've heard about 3D6. It's the murine version of bapineuzumab, essentially. And mice were dosed weekly with the antibody, and at the same time, they were dosed twice weekly with a drug called merimostat. Merimostat is an MMP inhibitor that inhibits MMP 1, 2, 3, 7, and 9, but it tends to have it has differing affinities for each of these, and it does have a higher affinity for 9 than some of the other MMPs. Key to this, merimostat does cross the blood-brain barrier. We did some preliminary studies to show that the twice-weekly dosing would maintain sufficient myrmestat levels in the brain, and those are the graphs that you see there. The plasma level drops quite quickly after dosing, but the brain level stays elevated up until 3 days, which is obviously why we chose the twice-weekly dosing of myrmestat. We also did monthly MRI imaging on these mice, and then post-mortem, we're working on the single-cell RNA-seq as well as an array of immunohistochemical analyses. So this is the only data I have to show you right now. We are still crunching data and doing stains and everything else, but preliminarily, MMP inhibition, I would say, had a minimal impact on microhemorrhage incidence. So what you see on the left part of the slide is our MRI data, and on the right part of the slide is our Prussian blue histology for microhemorrhages. So the first thing I'll say is that Prussian blue detects a lot more microbleeds than you see on MRI because they have to be a certain size in order for the MRI to detect them. But what you can see on the MRI, and these are just a few examples of the microhemorrhages that we saw on MRI, when we do the counts of microhemorrhages through MRI, we see absolutely no difference in the count number, whether we look at 3D6 alone or we look at MMP9 plus MMP inhibitor plus 3D6. So when it comes to larger bleeds, MMP9 inhibition doesn't seem to have a significant impact. However, when we look at Prussian blue, which you can see already the count numbers are much higher than the count numbers are by MRI. We looked at surface bleeds, so these would be leptomeningeal bleeds, and then we looked at parenchymal cortical microbleeds. And so what you can see is MMP inhibition with 3D6 is the blue bar, and then the red bar is 3D6 alone. And so we see maybe a 40 to 50% decrease in microhemorrhage number when it's assessed through Prussian blue staining. The reason, but this is still quite small numbers, we did have some attrition in the study, and so we're adding cohorts to the study, which is why we don't yet have that single cell data or anything else. We're waiting for all the animals so that we can go ahead and process all of that. So preliminarily, it looks like MMP inhibition may have a modest effect on microhemorrhages, but doesn't have a big effect on the larger bleeds that result from anti-A beta immunotherapy. So just to summarize, early studies in mouse models, as you heard from Dr. Lemire, predicted much of what we've seen in clinical trials. Rapid amyloid clearance is necessary for a benefit. Microglial-mediated amyloid clearance is necessary. Microhemorrhages were apparent early in the mouse models, and that has played out clinically, which we now call ARIA. Clinical trials indicate the earlier in disease progression we can get these therapies in, the greater the benefit and potentially the less risk of ARIA. Our single-cell RNA-seq studies looking at acute responses to antibody suggest microglia to microglia communication, but also microglia to peripheral immune cell communication is particularly increased following exposure to anti-A beta antibody. When we inhibit MMPs, we have a modest impact on microhemorrhages due to anti-A beta antibody in this mouse model, and we're continuing to add numbers to that study, and obviously we have a lot of outcome measures we want to look at there. So at IU, I just want to note that clinically we've treated 200 individuals now with monoclonal antibodies, the majority lecanemab. We're including 4 homozygotes when they pass all the other eligibility criteria. And currently our ARIA rate, I looked at the numbers a couple of weeks ago, our ARIA rate is running around 14%. So that seems to be quite positive. With that, I'll thank folks in my lab, some of whom are in the audience. I'll thank my IU collaborators and also my outside collaborators. And my postdoc, Kate Foley, is giving one of the speed talks, the 4-minute talks, about some of her other single-cell RNA-seq data. So please look out for that. Thank you for your attention.
Okay. Thank you very much, Donna. Our next speaker is Xavier Taylor from Eli Lilly and Company, and the title of his talk is Investigating the Cascade of Immunological and Vascular Events in Amyloid Immunotherapy.
Excellent. Well, thank you for the opportunity to speak and present and share this data with you today. As Cynthia said, investigating the cascade of immunological and vascular events in amyloid immunotherapy. These are my disclosures. So briefly, Alzheimer's disease, it's the most common form of dementia. It's characterized by the extracellular deposition of these parenchymal A-beta deposits, the intracellular accumulation of tau in the form of neurofibrillary tangles, and significant inflammation. Now, typically when we think about inflammation in the context of Alzheimer's disease, we typically think about activated astrocytes and activated microglia. However, as you've seen from these talks, there's also significant vascular pathology in the form of CAA. And the role of these alternative immune cells, such as these circulating immune cells, neutrophils, and monocytes, and meningeal immune cells like macrophages, dendritic cells, T cells, B cells, their role in actually interacting and associating with vascular pathology has been historically understudied. And now this is becoming increasingly even more important as A-beta immunotherapy, our most promising approach to modifying neurodegeneration, is going to be targeting both these parenchymal plaques as well as vascular plaques. Therefore, understanding the role of these immune cells, again, is increasingly more important. So we were actually the first to demonstrate that there's an engagement of these perivascular macrophages with these vascular amyloid immune complexes in our PDAPP mouse model. So as you've heard from earlier, we dosed with 3D6. So I prepared this video. Hopefully it plays. So X34, this is the vascular amyloid. This is taken from a penetrating vessel. 3D6 in green is going to come in and bind to that vascular amyloid, forming these vascular amyloid immune complexes. This is then followed by the engagement of CD169 positive perivascular macrophages in the cyan channel. And then finally, laminin is going to be in red. And this is a really nice marker because it helps to illustrate the anatomy and the physiology of the vessel. So when it stops playing, you can actually see here delineates really nicely where blood flow would be. So this is the lumen of the vessel. And then reflexive to this, you have these perivascular lymphatic drainage pathways. And in these drainage pathways, this is going to be where those immune interactions are taking place. So we've seen that macrophages are a pretty important cell type. However, we also wanted to characterize the other immune cells. And what we observed was that peripheral monocytes were highly recruited around these vascular amyloid deposits. So with our 3D6 treatment here, this is THIO-S, our vascular amyloid marker. GR1 represents our monocyte populations, and I just hope you can appreciate this really abundant increase in monocytes as well as this expansion of that perivascular space. So macrophages and monocytes are really important immune cells in this context, and we wanted to characterize that further. So this is from our most recent paper published late last year. And what we actually demonstrate is we can look and define the origins of these different cell populations. So MRC1, this denotes our tissue-resident macrophages, and CD74, these are going to be our monocyte-derived macrophages. The data that you're seeing is RNA-scope. So this is our 3D6 condition. Our amyloid is in yellow here. And I really like these markers because they help discriminate, again, the origins, and they segregate out pretty nicely. So here you can see this nice vascular resident macrophage that's staining with green, and then these other populations are CD74 positive. So both of these populations are going to be highly increased around the vessels after immunotherapy. We do see a slightly higher enrichment of these CD74 or these monocyte-derived populations, though. Importantly, characterizing further, we included a marker for TREM2. TREM2 is a really important gene. And in this context, you can see an increase in TREM2 coverage around these vessels and this nice colocalization or overlap staining with our vascular resident and our monocyte-derived populations. So given the clinical landscape, this is particularly important. Elector ran a INVOKE phase 2 clinical trial in which they used a TREM2 agonizing antibody for the treatment of Alzheimer's disease. Now, in this clinical trial, what they observed, they didn't see significant removal of amyloid, parenchymal, or vascular, but they did see a significant increase in microhemorrhaging in these patients, particularly those with the APOE4 allele. Therefore, kind of given the totality of this data, I think it's reasonable that the drainage of these TREM2 agonizing antibodies within these lymphatic compartments could be engaging with these TREM2 positive macrophages and maybe initiating this cascade of events. So a deeper characterization of our mouse model. You saw this image earlier. This is our PDAP mouse model. This is pearl staining, so this is essentially a stain for iron. This nice blue pattern here in the leptomeninges, this is where the majority of our microhemorrhages occur in our mouse model, roughly 90% to 95%. We do have some in the penetrating vessels as well, and we can use different markers like plasma protein extravasation markers. Fibrinogen, we mentioned that a little bit earlier. This is a nice marker to help show that blood-brain barrier breakdown at a more acute setting. It's also nice because we can use it to multiplex with other markers. So you can see here, this is our vascular amyloid and our 3D6 treated group. You see a lot of this fibrinogen being leaking from the vessel. What you also observe is when you look at vascular integrity, you see a really significant and robust loss of smooth muscle cells. And interestingly, where that muscle cell loss is occurring is where you see a lot of this fibrinogen. Endothelial cells, rather, they're fairly well preserved in the context of our treatments, and this actually tracks fairly well with the natural progression of CAA in patients, in which they also have a loss of smooth muscle cells, but a preservation of these endothelial cells. In the context of immunotherapy, we're accelerating this loss of smooth muscle cells. So a lot of the work that we've been doing has been characterizing tissue injury in these different stages. So phase 1, this is the earliest stage where you have a lot of this acute inflammation, fibrin, and thrombin activity. However, there's also this later phase of tissue injury, which denotes this reparative inflammation, angiogenesis, and tissue differentiation. So we wanted to utilize some new techniques and tools to actually understand these two different phases. So this is digital spatial profiling using nanostring. It's a really great tool, and it's allowed us to have a higher resolution of what's occurring at these vascular deposits. So essentially what this is, it's a multiplex hybridization assay. So we can have RNA probes or protein probes with these unique photocleavable linkers attached to them. We'll define our region of interest, a parenchymal plaque, or in our case, a vascular plaque. We'll illuminate that region of interest. This will cleave that barcode. We can aspirate it and get quantitative and semi-quantitative counts of either protein or RNA expression. So all the data that you see on the right, this would be from the protein panel. CD45, this is our most significantly differentially expressed protein, highly upregulated around these deposits. We have a loss of smooth muscle cells, which aligns nicely with some of the immunofluorescence data from earlier. A slight enrichment of CD31, these are your endothelial cells, so again, maybe some of that angiogenesis that could potentially be occurring. F480, our macrophage marker, highly enriched and upregulated around these deposits. MHC class 2, CD11B, this is antigen presentation machinery that's also occurring around this site. CTLA4, this is a T-cell inhibitor protein, so less T-cell inhibition would correlate with more T-cell activity. And in line with that, we have this increase in granzyme B and CD28, both activated T-cell markers. Finally, fibronectin, so this is a fibroblast marker, and it's near on the same level as enrichment as our macrophage populations. So you can see on the right here, a lot of fibroblasts and fibrotic activity occurring around these vessels. So using these different protein markers, we can actually look and see how they correlate with one another. And what we observed was macrophages were highly correlated to fibroblasts, only in the context of our 3D6 treatment. So the more macrophage activity you have, the more fibroblast activity. What was interesting, and I was actually kind of expecting this, but we see that macrophages were not correlated to smooth muscle cell loss, and this was true neither for the IgG or the 3D6. What we observed was actually correlated to smooth muscle cell loss were these peripheral infiltrating immune cells. So CD28, the more CD28 you have, the more smooth muscle cell loss you have. This was significant both for the IgG and in the 3D6. We're just exacerbating this loss in the context of our immunotherapy. True again for our monocyte populations, the more infiltrating monocytes we had, the more smooth muscle cell loss you had. This was significant in the context of 3D6 and near significant in the context of IgG. So a little bit interesting here, we don't quite understand the directionality at this point. We don't know if a loss of smooth muscle cells facilitates the infiltration of these immune cells, or by virtue of these immune cells infiltrating, they're actually causing the smooth muscle cell loss, but we're actively working to uncover that. Finally, this is the gene expression panel. So this has been unpublished. This is your guys' first time seeing this. And this is from the 1,000-plex RNA panel that we ran. We ended up with around 300 transcripts per cell, which is a really nice enrichment. And I just hope you can appreciate all these macrophages around this vascular event. So MS4A7, this represents our macrophage populations, and again, highly upregulated around these vascular events. We can get separation of our different populations, so our PVM1s and PVM2s, these represent our monocyte-derived versus our vascular resident populations, and we can use different algorithms to see how these immune cells actually talk to one another. What we observed was that these fibroblasts are highly responsive in talking to our vascular resident populations. We're working to uncover what they're saying. But we also see that our monocyte-derived population talks quite a bit to smooth muscle cells. Our monocyte-derived population is becoming increasingly more important. We want to see their gene expression. TREM2 is a gene that they're upregulating quite a bit. As you heard earlier, complement system, we do see a complement system genes involved in this process, but our most significantly differentially expressed pathway were these extracellular matrix remodeling genes. So this has been a really nice tool. We can correlate and help understand how our ARIA mouse model is overlapping with human CAA patient data. And what we observe is that a lot of our pathways do overlap, and these pathways correspond to, of course, complement, phagocytosis, and antigen presentation. So this is our proposed hypothesis behind how these mechanisms of microhemorrhage are occurring, at least in the context of our mouse model. So first and foremost, we have our immune activation and our immune complex formation, so our antibodies binding to that vascular amyloid, initiating this cascade of immune responses. Our macrophages are going to be the first responders, and we think after this they secrete all these chemokines and cytokines that help recruit all these peripheral immune cells to that site of damage. That's going to correspond with plasma protein extravasation, vascular dysfunction, and maybe some concomitant wound healing and repair also occurring. But ultimately, this process yields cerebral vascular damage and microhemorrhaging with this vascular permeability. So this is our current idea, and we're actively exploring this pathway and these mechanisms a little bit more. So finally, these are my acknowledgments. Big thank you to all the members that helped make this project go forward and be publishable so we can present to you today. Thanks to Pascal's lab for a lot of the digital spatial profiling and Ron's lab for providing reagents and mouse models. So with that, I'd be happy to answer any questions.
So we have this presentation is open for questions. Please go to the microphones, which are labeled. All right, microphone 3. Go ahead.
Aaron Cantor, Thrini Bio. Thank you all for your talks. So a question for Xavier. So there are TREM2 intermediate and TREM2 high populations of macrophage and microglia, and they respond differently to some of the anti-TREM2 antagonists. And I was just wondering, were you able to distinguish between those subsets? And do you think there's any consequence of that?
We weren't able to discriminate at this point. We just used RNA-scope to really profile and know
Microphone close to them, yeah.
Sorry. We weren't able to discriminate at that point. We're just doing RNA-scope, so we know that they're TREM2 positive, but in the future, I think it would be worth exploring that a little bit further to see if we can parse what populations are more TREM2 high versus TREM2 low.
They respond differently to drugs. That's why I am interested, so thank you.
Yes.
Go ahead. Identify yourself.
Dave Morgan, Michigan State University. Beautiful talks. I didn't see all of them, and I have a question because I'm scared to death by what Dr. Taylor just told us. Does this loss of smooth muscle cells eliminate vasogenic regulation? And because of that, are we going to see a whole bunch of people who are going to get worse after a certain period of time because they don't have the right blood flow? This really concerns me. And maybe Delphine can add to that. Maybe you've seen something pathologically.
We've done the smooth muscle actin immunohistochemistry in our immunized cases, and yes, as we see increase of the vasculopathy, we see decrease of the smooth muscle actin, so it's kind of fit with Xavier finding. But I agree, we don't know what is cause or what is consequence, but there is less SMA after immunotherapy. Yeah.
Number 7.
Thank you, Bruce Brew from Sydney, Australia. 2 issues. None of the models seem to explain what actually happens in humans in terms of the temporal sequence of ARIA. So it seems to go up in the first few months, peaks, and then decreases. and it decreases far before the actual amyloid clearance at 1 year, 1.5 years. So what's the explanation for the drop? And second, if primed macrophages, provascular macrophages and astrocytes are important, then you'd expect ARIA to be perhaps more frequent in HIV patients who have Alzheimer's.
So I think the reason that it's occurring very early is you have a system that's already weak. You have vessels that have been degrading for a long period of time. You could already have some smooth muscle cell loss, and then you exacerbate that early. But as you saw, there is some fibrotic remodeling, some wound healing and repair that's also occurring. So I think that given a sufficient amount of time, when you have those fibrotic responses, collagens, laminins, and all these extracellular matrix remodeling proteins remodel that space, that can be protective in the long run.
Thank you. Number 5.
Yes, Steve Rich from Com Pharmaceuticals. A question, it was alluded to that the similarities between this and CAA-related inflammation. And there's a clinical disparity because when we see CAA-related inflammation, we're often treating these patients with very high-dose steroids for a very long time with really pretty poor outcomes. Yet when we're using these anti-beta amyloids, the recommendations are things such as watch them again in a month, stop the infusion, maybe skip a dose, and there's a disproportion here. Do we know the outcomes in detail from people who've gotten this anti-amyloid-induced ARIA versus what is CAA-related inflammation?
Yeah, so I think it's important to point out that typically in asymptomatic ARIA cases, our approach clinically is to pause dosing, monitor for recovery, and if the ARIA is recovered, a lot of times those patients are offered the option to reinitiate treatment. But when we have symptomatic ARIA, especially severe symptomatic ARIA, which we've had a couple of symptomatic ARIA cases in Indiana, that is treated with steroid as well as just monitoring. So we do treat with dexamethasone and also when they're severely symptomatic, they're not given the option to restart any therapy afterwards. We've had some individuals who have developed asymptomatic ARIA. That ARIA has resolved, they've restarted treatment, and they've redeveloped ARIA. And in those cases, they do not pursue any more treatment after that. If it restarts once, that's it. I think the relationship between CAA-RI and anti-beta immunotherapy related ARIA, I think there's still a lot we need to understand about mechanisms of CAA-RI. I don't think we have the data at hand to compare the severity of the inflammatory response in CAA-RI directly to the inflammation that we see with anti-beta immunotherapy. It may be that those individuals with CAA-RI are having a much more exacerbated inflammatory response than we're inducing with anti-beta antibody. I'm not sure if anyone wants to comment.
I think we better move on because we have number 2.
Just answer one question. Is there a database of these iatrogenic cases and their outcomes?